endogenous and exogenous damage. Types of endogenous damage. The various damages caused to DNA due to endogenous cellular process are A. Oxidation of bases, B. Alkylation of bases, C. Hydrolysis of bases, D. Adduct formation, E. Mismatch of bases. Causes of exogenous damage. UVA and UVB radiations, ionizing radiations, thermal disruption, industrial chemicals such as vinyl chloride, hydrogen peroxide, polycyclic aromatic hydrocarbons, alkylating agents. DNA repair systems. DNA damage occurs spontaneously and as a result of ubiquitous environmental agents. Most organisms possess some capacity to repair the DNA, and DNA is the only macromolecule which is repaired by cells. The repair mechanisms are divided into three categories: damage reversal, damage removal, and damage tolerance. Damage reversal. Photoreactivation. Cells are endowed with repair mechanisms to correct radiation damages and to bring the DNA back to its normal state under the influence of intense blue light. This reversal of the mutagenic effect of UV radiation by visible light is called photoreactivation. The photolyase enzyme catalyzes this reaction, which utilizes light energy to break the bonds between purine dimers. Here you can see the action of photolyase along with white light, which breaks the photodimer into separate thymine molecules. But on exposure to UV light, the thymine residues in turn become a photodimer. Ligation of single strand breaks. Single strand breaks or SSBs arise from direct attack by intracellular metabolites and from spontaneous DNA decay. Simple breaks in one strand are rapidly repaired by DNA ligase. Microbial mutant lacking ligase tend to have high levels of recombination since DNA ends are recombinogenic. Bloom syndrome in humans is involved with DNA ligase deficiency. Let us now study about damage removal type DNA repair mechanisms. To compensate for the many types of DNA damage that occurs, cells have developed 5 major DNA repair mechanisms, which are base excision repair or BER, mismatch repair or MMR, nucleotide excision repair NER, and double strand break repair, which includes both homologous recombination HR and non-homologous end joining NHEJ. In this table, you can see the 5 different types of DNA repair mechanisms and the important genes which are involved in the same. Base excision repair or BER. Base excision repair is primarily responsible for removing small non-helix distorting base lesions from the genome. BER is initiated by DNA glycosylases, which recognize and remove inappropriate bases forming apurinic site or AP site and are cleaved by an AP endonuclease. DNA polymerase then removes the damaged region using its 5' to 3' exonuclease activity and correctly synthesizes the new strand using the complementary strand as a template. The resulting single strand break can then be processed by either short patch or long patch base excision repair method. Short patch method. In short patch method of BER, a DNA glycosylase cleaves the bond between the nucleotide base and ribose, leaving the ribose phosphate chain of the DNA intact, but resulting in an apurinic site. 8-oxoguanine DNA glycosylase 1 or OGG1 removes 7,8-dihydro-8-oxoguanine or 8-oxo-G, one of the base mutations generated by reactive oxygen species. Polymorphism in the human OGG1 gene is associated with the risk of various cancers such as lung and prostate cancer. Uracil DNA glycosylase, another base excision repair enzyme, excises the uracil, that is the product of cytosine deamination, thereby preventing the subsequent cytosine to thymine point mutation. N-methylpurine DNA glycosylase or MPG is able to remove a variety of modified purine bases. The AP sites in the DNA that result from the action of BER enzymes are repaired by AP endonuclease 1 or APE1. APE1 cleaves the phosphodiester chain 5' to the AP site. The DNA strand then contains a 3' hydroxyl group and a 5' A-basic deoxyribose phosphate. DNA polymerase beta or Pol beta inserts the correct nucleotide based on the corresponding WC pairing and removes the deoxyribose phosphate through its associated AP lyase activity. The presence of X-ray repair cross-complementing group 1 or XRCC1 is necessary to form heterodimer with DNA ligase 3 LIG3. XRCC1 acts as a scaffold protein to present a non-reactive binding site for Pol beta and brings the Pol beta and Lig3 enzymes together at the site of repair. Poly-ADP ribose polymerase or PARP1 interacts with XRCC1 and Pol beta and is a necessary component of the base excision repair pathway. The final step in the repair is performed by Lig3, which connects the deoxyribose of the replacement nucleotide to the deoxyribosyl phosphate backbone. This pathway has been named short patch BER. Long patch base excision repair. Long patch base excision repair replaces a strand of nucleotides with a minimum length of 2 nucleotides. Long patch BER requires the presence of proliferation cell nuclear antigen or PCNA, which acts as a scaffold protein for the restructuring enzymes. Other DNA polymerases, possibly Pol delta and Pol epsilon, are used to generate an oligonucleotide flap. The existing nucleotide sequence is removed by flap endonuclease 1 or FEN1. The oligonucleotide is then ligated to the DNA by DNA ligase 1 or Lig1, sealing the break and completing the repair. Nucleotide excision repair or NER. Nucleotide excision repair is the most flexible of the DNA repair pathways, considering the diversity of DNA lesions it acts upon. The most significant of these lesions are pyrimidine dimers caused by the UV component of sunlight. Other NER substrates include bulky chemical adducts, DNA intrastrand crosslinks, and some forms of oxidative damage. The common features of lesions recognized by the NER pathway are that they cause both a helical distortion of the DNA duplex and a modification of the DNA chemistry. Mammalian nucleotide excision repair or NER eliminates carcinogen DNA adducts by double endonucleolytic cleavage and subsequent release of 24 to 32 nucleotide long single stranded fragments. The NER process requires the action of more than 30 proteins in a stepwise manner that includes damage recognition, local opening of the DNA duplex around the lesion, dual insertion of the damaged DNA strand, gap repair synthesis, and strand ligation. There are two distinct forms of nucleotide excision repair: GGNER, which is the global genomic NER, which corrects damage in transcriptionally silent areas of the genome, and TCNER, which stands for transcription coupled NER, which repairs lesions on the actively transcribed strand of the DNA. These two sub-pathways are fundamentally identical except in their mechanism of damage recognition. In GGNER, the XPC or Xeroderma pigmentosum complementation group C protein complex is responsible for the initial detection of damaged DNA. Conversely, damage recognition during TCNER does not require XPC, but occurs when the transcription machinery is stalled at the site of injury. The stalled RNA polymerase complex is displaced in order to allow the NER proteins to access the damaged DNA. This displacement is aided by the action of CSA or cocaine syndrome A and CSB proteins as well as other TCNER specific factors. The subsequent steps of GG and TCNER proceed in an essentially identical manner. XPA and the heterotrimeric RPA or replication protein A then bind at the site of injury and further aid in damage recognition. Next, the XPB and XPD helicases, components of the multi-subunit transcription factor TF2H, unwind the DNA duplex in the immediate vicinity of the lesion for the enzyme RNA polymerase II to begin transcription. The endonucleases XPG and ERCC1, that is Excision repair cross complementing group 1 or XPF, then cleave one strand of the DNA at positions 3 and 5 to the damage respectively, generating an approximately 30 base oligonucleotide containing the lesion. Finally, the nick in the repaired strand is sealed by a DNA ligase, thus completing the nucleotide excision repair. 3 syndromes are associated with inborn defects in nucleotide excision repair. They are Xeroderma pigmentosum or XP, cocaine syndrome or CS, and trichothiodystrophy, which is also known as TTD, all characterized by exquisite sun sensitivity. The disorder arises from mutations in one of the 7 genes, that is XPA to XPG. Cocaine syndrome caused by mutation in the CSA or CSB genes is a TCNER specific disorder that is remarkably dissimilar from Xeroderma pigmentosum. No predisposition to cancer is observed because TCNER defect causes CS cells to be particularly sensitive to lesion induced apoptosis, thereby protecting against tumorigenesis. Physical and neurological developments are impaired, resulting in dwarfism and dysmyelination. The syndrome includes features of premature aging, which is related to the increased trigger for apoptosis induced by transcriptional arrest from endogenous lesions in combination with the TCNER defect. TTD is a condition sharing many symptoms with the cocaine syndrome, but with the additional hallmarks of brittle hair, nails and scaly skin. TTD is not associated with cancer. Mutations in the XPD or XPB genes gives rise to all 3 diseases. Thus, as subunits of TF2H, XPB and XPD have dual functions, NER and transcription initiation. Mutations not only compromise NER, but also affect transcription, causing developmental delay and reduced expression of the matrix proteins that causes brittle hair and scaly skin. TCNER initiates when RNA polymerase stalls at a lesion in DNA. The blocked RNA polymerase serves as a damage recognition signal which replaces the need for the distortion recognition properties of the XPC RAD23B and DDB complexes. CS proteins such as CSA and CSB bind some types of DNA damage instead of XPC RAD23B. Mutations in TCNER machinery are responsible for multiple genetic disorders, including TCNER associated diseases such as trichothiodystrophy or TTD is an autosomal recessive inherent disorder characterized by brittle hair and intellectual impairment. Cocaine syndrome, also known as Neal-Dingwall syndrome, is a rare autosomal recessive neurodegenerative disorder characterized by growth failure, impaired development of the nervous system, abnormal sensitivity to sunlight or photosensitivity, eye disorders and premature aging.